So my name is Alex Sossick. So I work at the Gurdon Institute, which is part of the University of Cambridge, and we're a developmental biology and cancer research institute funded by the Wellcome Trust and Cancer Research UK. And I run the imaging facility, so basically the advanced microscopes in the institute, such as confocal microscopes, super resolution, and other advanced techniques that people want to use. And we adapt to people's need. It's a course aimed at people using especially light microscopy in the cell biology field. We're aiming at PhD students and postdocs who are either biologists or physicists who want to develop a better understanding of modern microscope techniques. The things we're looking for are how much of a benefit it would be, and also whether they're in a position where they'll take knowledge back to the host institute. The advantage to biologists is they get to use these advanced techniques, but also learn from physicists how the techniques work. So with a better understanding, they can get better results from the microscopy that's available to them. And for physicists, then it means that they can learn about the biology and understand what people actually want to image and learn the kind of imaging requirements of the biologists. And we're aiming to teach both the fundamentals of modern microscopy. The course is structured in two halves. The first half is basic microscopy, learning the fundamentals of how to set up a microscope. So color illumination, DIC, phase contrast and fluorescence. And then the second half of the course is more advanced techniques like light sheet, confocal, multiphoton, and whatever the latest techniques like super resolution microscopy are covered. As far as the practicals go. And then we have a really excellent array of lecturers on the course covering all sorts of microscopy topics and techniques. We basically try and find the best people we can to give talks, and we try and give as broad a knowledge of light microscopy as we possibly can to the students. So this year's course has varied slightly from last year. Clearly we have the same formula, but we've got a bit more image analysis included towards the end of the course. The course evolves every year, there's different techniques covered, and we always bring in a few new lecturers and as techniques become more interesting. So we try and adapt the course every year to the latest trends and what we think students should be learning or would want to learn. I became involved in organising the course a long time ago when the first course basically started. I thought it was a very important course because if you have the fundamental knowledge of microscopy and biology, then you can really push the techniques you're able to use. So you're not just using one microscopy technique, you're able to use lots of different techniques, which means that you're able to do a lot more experiments. I'm very keen on teaching people how to use different techniques, and the course is great for that. So what I enjoy about organising the course is trying to pass on a lot of knowledge to students, not just my knowledge but other people's knowledge, and trying to create an environment where it's really ideal for learning as much as you possibly can. Partly from the breadth of lecturers that we have, but also from the companies that come. We try and make it so that it's a very friendly environment, so people are learning all the time and trying to gain new techniques. And I think that's very important as a skill, and it's something I really enjoy, creating the environment and passing on knowledge. Scientifica helps the course in a number of ways. Importantly, Scientifica brings equipment, so it's brought its multiphoton microscope this year. It exposes students to multiphoton microscopy, and we're able through this to teach students how to best get results from multiphoton microscopy, how to set up a multiphoton microscope and learn the techniques that are possible, and get tips and tricks for their own work when they go home to their host institution. Secondly, Scientifica provides personnel to actually support the microscope that they've brought. Scientifica are able to pass on skills that they've learned to students and really appreciate the fact that companies come to the course and are willing to pass on knowledge to the students and really interact with the students and become part of the faculty of the course. Thirdly, Scientifica provides sponsorship for the course. And as we've found other companies, this sponsorship goes towards providing students with some bursaries so that we're able to get students on merit. We're that aren't necessarily able to afford to come to the course, which is vital. And we're also able to pay for lecturers to come to the course from the sponsorship, which means that we're actually able to get the best lecturers available. We wouldn't be able to do that without the sponsorship that companies like Scientifica provide, so that we're able to, again, to provide the course at the level that we want.